Ready to mark yourself a star? Let's use the checklist the smart way. Ask, answer, and tick. This is the official self-evaluation checklist straight from the Cambridge IGCSE Biology Coursebook. Chapter 13, it's all about excretion and homeostasis. Section 13.1 focuses on the kidneys and how waste is removed. Section 13.2 covers homeostasis, hormones, and temperature regulation. Let's begin. There are 13 points to check. Section 13.1, Excretion and the Urinary System. 1. Can I state where carbon dioxide, urea, excess water, and ions are excreted? Number 1. Carbon dioxide. Excreted by the lungs. Produced during aerobic respiration, removed from the blood in the alveoli and breathed out. Number 2. Urea. Excreted by the kidneys in urine. Formed in the liver from excess amino acids, deamination. Number 3. Excess water. Mainly excreted by the kidneys in urine. Also lost through the skin as sweat and the lungs as water vapor. Number 4. Excess ions, salts. Excreted by the kidneys in urine. Also removed through sweat glands in the skin. Here is the checkpoint number 1. It is 2. Can I identify the kidneys, ureters, bladder, and urethra on a diagram? This is a diagram you need to spot. This is kidneys. 2 bean-shaped organs located on either side of the spine. Here inside the kidneys. 3 things done you have to remember. Number 1, ultrafiltration. Number 2, selective reabsorption. And number 3, urine formation and excretion. These are ureters, thin tubes that carry urine from each kidney to the bladder. 1 for each kidney. Its function is to carry urine from kidneys to urinary bladder. This is the urinary bladder, muscular sac-like organ that stores urine, positioned in the lower abdomen in the center. And this is urethra, a short tube leading from the bladder to the outside of the body. It is also a carrier, but here it carry urine outside the body. Ureter and urethra both are carriers. Ureter equals enters bladder, urethra equals exits body. 3. Can I outline the structure of a kidney and nephron with blood capillaries? Regarding kidney structure, each kidney has 2 main internal regions: cortex, outer region where filtration begins; medulla, inner region with loops of nephrons and collecting ducts. Leading from is a tube called the ureter. Her is the pelvis. It is the site of collection of all nephrons to form the ureter, so don't miss cortex, medulla, pelvis. Regarding nephron structure, each nephron starts in the cortex, where it forms a Bowman's capsule around a glomerulus, a bundle of capillaries. Here is the Bowman's capsule, and this is the glomerulus. This is the proximal convoluted tubule or coiled tubule. This is the loop of Henle. This is the distal convoluted tubule, and this is the collecting duct which go to the ureter. Capillaries wrap around the nephron for reabsorption, which bring blood in through a branch of the renal. Filter it in the glomerulus. Reabsorb useful substances. Carry away filtered blood via a branch of the renal vein. This structure enables, first, filtration in the glomerulus. Second, selective reabsorption along the tubule. Third, urine formation, passed to the ureter. Number 4. Can I describe how urea is made from deamination in the liver? Urea is formed in the liver by breaking down excess amino acids in a process called deamination. The following is happen. 1. Protein from food is digested in the stomach and small intestine, producing amino acids. 2. These amino acids are absorbed into the blood capillaries in the villi of the small intestine. 3. The blood carries them to the liver via the hepatic portal vein. 4. In the liver. Some amino acids are used for protein synthesis or released into the blood. Excess amino acids are not stored. Instead, they are broken down in a process called deamination. The energy-containing part is turned into carbohydrate. The nitrogen-containing part is converted into urea. This step is called deamination. 5. Urea is then released into the blood plasma, kidneys, and excreted in urine. So deamination removes the nitrogen-containing amino group from the amino acid. This forms ammonia, which is toxic. The liver quickly converts ammonia into urea, which is less harmful. Urea is then released into the bloodstream, filtered by the kidneys, and excreted in urine. In summary, excess amino acids, deamination, ammonia, urea, excreted in urine. 5. Can I describe how filtration and reabsorption happen in a nephron? Number 1. Filtration. Blood enters the glomerulus, a ball of capillaries in the kidney cortex. High pressure forces small molecules like water, glucose, ions, and urea out of the blood and into the Bowman's capsule. This process is called ultrafiltration. Large molecules like proteins and blood cells are too big to pass through the filter and remain in the blood. Number 2. Reabsorption. As the filtrate flows along the nephron tubule, useful substances are selectively reabsorbed into the surrounding blood capillaries. These include all of the glucose, most of the water, some of the ions depending on the body's needs. This process ensures that only waste products, like urea, excess water, and excess salts, are excreted. The remaining liquid, now called urine, flows into the ureter, then to the bladder for excretion. Key concepts. Number 1. Filtration occurs in the glomerulus. Number 2. Reabsorption happens along the nephron tubule, especially in the proximal convoluted tubule. Number 6. Can I describe how urine is formed and what it contains? Urine formation happens in 3 main steps inside the nephrons of the kidneys. See this photo here also. First, filtration. Blood enters the glomerulus under high pressure. Small molecules such as water, glucose, urea, and ions are forced into the Bowman's capsule. Large molecules, e.g. proteins, blood cells, stay in the blood. 2. Selective reabsorption. As the filtrate flows through the nephron tubule, all glucose, most water, and needed ions are reabsorbed into the blood. Urea and excess water and salts are left behind. Third, formation of urine. The final liquid, now called urine, is made of urea, a toxic waste from amino acid breakdown. Excess water, excess salts, or ions. It flows into the ureter, then to the bladder, and is excreted through the urethra. Urine does not normally contain glucose, proteins, or blood cells. If present, it's a sign of disease, e.g. diabetes or kidney damage. Now it is section 13.2, about homeostasis. 7. Can I define homeostasis? Homeostasis is the regulation of internal conditions to maintain a stable internal environment, despite changes in the external environment. In short, homeostasis equals keeping internal conditions constant, like keeping a constant temperature of around 37 degrees Celsius, keeping a constant amount of water, keeping a constant concentration of glucose. It's your body's thermostat plus autopilot combined. 8. Can I describe the effect of insulin? Insulin is a hormone made by the pancreas, beta cells. It is released when blood glucose concentration rises, example after eating, tells liver and muscles, store excess as glycogen. When the concentration of glucose in the blood goes up, the islets in the pancreas detect this and secrete insulin into the blood. When insulin reaches the liver, it causes the liver to absorb glucose from the blood. Excess glucose is converted into the insoluble polysaccharide called glycogen. This is stored in the liver. In short, insulin reduces blood glucose by promoting its uptake and storage as glycogen. 9. Can I explain how negative feedback keeps values close to a set point? Yes. Negative feedback is a process the body uses to keep conditions such as blood glucose concentration close to a normal level. This normal level called the set point. When the level of a variable, like blood glucose, moves outside this set point range, either too high or too low, receptors detect the change, and the body responds by activating mechanisms to reverse the change and bring the value back to normal. For example, If blood glucose rises above the set point, as after eating sugary food, cells in the pancreas detect this and secrete insulin. Insulin causes the liver to absorb glucose from the blood and convert it into glycogen, lowering the blood glucose level. If blood glucose falls too low, as after skipping a meal and exercising, the pancreas secretes glucagon, which causes the liver to break down stored glycogen into glucose, raising the blood glucose level. So this is a negative feedback loop. Change happen, detect by sensor, response happened, values back to normal again. Again, never forget, negative feedback keeps internal conditions stable by detecting deviations from the set point and triggering corrective responses. 10. Can I describe how liver and pancreas control blood glucose? Liver is the glucose warehouse. Pancreas is the hormone manager. The pancreas monitors blood glucose concentration. When glucose levels move outside the normal range, called the set point, the pancreas responds by secreting hormones that signal the liver to correct the imbalance. If blood glucose is too high, for example after eating a meal, the islets in the pancreas detect this and secrete insulin into the blood. Insulin signals the liver and muscle cells to absorb glucose from the blood. The liver converts glucose to glycogen, a storage carbohydrate, and stores it. This reduces the glucose concentration in the blood back toward normal. If blood glucose is too low, for example after fasting or exercise, the pancreas detects that and secretes glucagon. Glucagon signals the liver to break down stored glycogen into glucose. The liver releases this glucose into the blood, increasing blood glucose concentration. This system allows the body to maintain blood glucose within a narrow range and is an example of negative feedback, where the liver and pancreas work together to bring conditions back to normal when they change. Eleventh, can I identify structures of the skin in a diagram? To identify skin structures in a diagram, like that one in your coursebook, you need to label the key parts involved in protection, sensation, blood supply, and temperature control. This is sweat pore, opening on the surface where sweat is released. Sweat duct, tube that carries sweat to the surface. This is sweat gland, coiled gland that produces sweat. Hair extends from the follicle through the skin surface, and this is hair erector muscle, contracts to pull hair upright, helps trap heat. Now coming to blood vessels. This is arteriole, it is small artery, brings oxygenated blood. And this is venule, it is a small vein, carries deoxygenated blood away. Capillaries connected them, allow heat transfer near the skin surface, and here one another important connection between them. It is shunt vessel, uses surface capillaries to reduce heat loss. Now there is some receptors. Temperature receptor, detects changes in external temperature. Pressure receptor, detects mechanical pressure. Sensory neuron, transmits signals from receptors to the central nervous system. Another structure you must identify in skin is fat cells, adipose tissue. It provides insulation and energy storage. Lastly, twelfth, can I describe how the hypothalamus controls temperature regulation? The hypothalamus is a part of the brain that acts as the body's internal thermostat. It contains temperature receptors that detect the temperature of the blood flowing through it. It also receives impulses from skin temperature receptors that detect external temperature changes, as cold air or water. When the hypothalamus detects a temperature below or above the set point, approximately 37 degrees Celsius, it sends nerve impulses through motor neurons to effectors like number 1, sweat glands, number 2, blood vessels in the skin, number 3, skeletal muscles, for shivering. When body temperature is too high, above 37 degrees Celsius, sweat glands are activated, produce more sweat. Evaporation cools the body. Blood vessels in the skin undergo vasodilation. More blood flows through the shunt vessels near the surface. Heat is lost by radiation. Erector muscles relax. Hair lies flat so no insulating air layer is trapped. Skeletal muscles remain relaxed. No shivering occurs. When body temperature is too low, below 37 degrees Celsius, sweat glands are inhibited, sweating stops, reduces heat loss. Blood vessels in the skin undergo vasoconstriction. Less blood flows through the shunt vessels near the surface, less heat lost by radiation. Heat is conserved. Erector muscles contract, hair stands, 13. Traps insulating air layer. Can I describe how insulation,
This feedback
sweating,
system helps
shivering,
maintain a stable core
vasodilation,
temperature
and
around
vasoconstriction
37
work?
degrees Celsius. An
First,
example of
describe
homeostasis.
insulation. The fatty layer under the skin, the adipose tissue, traps heat and slows heat loss. In cold conditions, erector muscles contract, causing hairs to stand up, trapping a layer of warm air near the skin, and this is an insulating effect. Second, describes sweating. When the body is too hot, sweat glands release sweat onto the skin surface. Evaporation of sweat uses heat energy from the skin, cooling the body. In cold conditions, production stops to conserve heat. Third, describes shivering. When body temperature drops, skeletal muscles contract rapidly and repeatedly. This generates heat from respiration, warming the blood and body. Fourth, describe vasodilation. When too hot, arterioles near the skin widen, dilate. More blood flows to surface capillaries, allowing more heat to be lost by radiation to the environment. And lastly, describe vasoconstriction. When too cold, arterioles near the skin narrow, constrict. Less blood reaches the surface capillaries, reducing heat loss from the skin. Each of these 5 responses is coordinated by the hypothalamus and helps maintain body temperature near 37 degrees Celsius, an example of homeostasis. Well done. If you understood these key ideas and can recall the right keywords, an A star is very near. Keep going. The more boxes you tick, the closer you get. Don't forget to review, stay confident, and move on to the next chapter. You've got this.